This video will focus on the process of polymerase chain reaction, or PCR. So let's start with the basics. PCR is a technique used to amplify DNA fragments. The goal of it is to take a little bit of DNA and make a lot of DNA. In essence, PCR is in vitro, that is, in a test tube, replication of DNA. So, for genes or other DNA that we're interested in, we can now use this technique to make a bunch of copies of that region and are not reliant on the amount of DNA in the original sample. To accomplish PCR, we need five basic components. The first is template DNA, which is the sample of DNA we intend to amplify part of. Secondly, we need primers. Primers help us target specific regions of DNA and are made up of short, single-stranded DNA sequences. Next, we need a DNA polymerase enzyme. From DNA replication, we know that DNA polymerase is the main workhorse enzyme for the process, and we will use a DNA polymerase from a prokaryote that has a heat-stable version of the enzyme. Next, we need the building blocks for our new strands of DNA. These are called dNTPs and are the four individual nucleotides in our DNA with a triphosphate form. Lastly, we need a buffer that maintains concentrations of particular salts like magnesium and proper pH for the PCR process to commence. Once we have assembled our ingredients in a tube, we are ready to load them onto a thermal cycler, which will take us through the three-step process of amplification. The first step in this process is denaturation, which happens at high temperatures and acts to separate the double-stranded DNA into single strands. Note that in DNA replication in our bodies, we employ enzymes like helicase to unwind the DNA. But in PCR, we simply use heat. This high heat is the reason we need a heat-stable DNA polymerase. The second step of PCR is the primer annealing step. Once the strands are separated, the samples are quickly cooled to allow the primers to bind or anneal. These primers bind to the template DNA in regions where they are complementary, which allows us to design primers to target specific regions in our genomes. Finally, the last step is elongation, also called extension. Once the primers bind, the temperature is raised slightly to promote DNA polymerase binding and the synthesis of complementary DNA strands. So, in the example here, our single strand of DNA is now two strands. Then we simply repeat. PCR is done over many cycles, often 25 to 35 times, with each cycle doubling the amount of DNA. Each cycle contains the three steps: denaturing, annealing, and elongation. Repeating these steps over and over results in an exponential increase in our DNA and turns a little bit of DNA into a lot of DNA.